Hi folks, and welcome to AP Biopharma with Dr. D. This is part 5 of this series, How Structure Determines Function, a Tale of Biomolecules. In this part, we're going to be talking about proteins. So proteins are also called polypeptides. They're called polypeptides because they're polymers of amino acids. And proteins are polymers of actually 20 different amino acids. And the amino acids can be arranged in any order. Imagine how many different polypeptides you can make using 500 amino acids. The order of amino acids will determine the structure of the protein, and the structure, as you know, determines the function. So you can build, just with 20 amino acids, a great variety of different structures and therefore functions. The order of the amino acid is also written in your genes, in the DNA. So let's examine some functions of proteins. And the functions of proteins are grouped in basically several large groups. And I used to say to my 9th grade freshman biology students that if they can't remember what proteins do, they should simply say proteins do everything. And they really do. Proteins are the workhorses of the cell. What you're most familiar with probably is enzymatic proteins. Proteins are enzymes. They function by specifically accelerating, speeding up chemical reactions. So they control chemical reactions inside a cell. And an example would be digestive enzymes, which catalyze the hydrolysis of bonds in our food. A second group of functions is proteins function as defensive proteins. They defend organisms against pathogens. Pathogens are either parasites or viruses or bacteria that can potentially harm the organism. And there are many different defense proteins, but the best example I have for you are antibodies. Antibodies are produced by our immune system to help inactivate and destroy viruses and bacteria. Transport proteins is the next group. Proteins are can be part of the cell membrane of cells and help transport substances in and out of a cell. Proteins can also transport substances throughout an organism. And an example, the best example really is hemoglobin. Hemoglobin is an iron-containing protein that will transport oxygen to every cell and every tissue in your body. Another group are storage proteins. This is a group we normally don't think much about. So proteins can store amino acids. They can be a source of amino acids. Why do organisms need amino acids? Well, they need amino acids to build their own proteins, and also in the process of breaking down the protein to get those amino acids, organisms can extract energy. So for instance, casein in the protein of milk is a major source of amino acids for a baby. Plants have storage proteins in their seeds to provide energy for the growing seed, for developing embryo. Ovalbumin is the protein in egg white, and it is used as an amino acid source for the developing embryo. Hormonal proteins. Their function is to coordinate an organism's activity to maintain homeostasis. Hormonal proteins are usually produced by one part of the body, by a gland, and then function in a different part of the body as part of the endocrine system of mammals. An example would be insulin. Insulin is a hormone which is secreted by the pancreas and would cause other tissues to take up glucose, thus lowering blood glucose concentration. Now, there's signaling proteins, such as hormonal proteins. There's also receptor proteins. Those are the proteins on cell surface whose function is to receive signals from the environment and transmit them inside the cell and to elicit a response, essentially. An example shown here are receptor proteins which are built into the membrane of nerve cells, and they can detect signaling molecules released by other nerve cells. So they're involved in neuronal signaling. Structural proteins. Structured proteins provide structure and support. The internal support of cells is made out of proteins. However, the most famous structured protein for us is keratin. Keratin is the protein that makes up hair. Our hair is made up of keratin. Keratin additionally is used to make up horns, feathers, and other skin appendages. Insects like spiders can use silk fibers to make their cocoons and webs respectively. These are also proteins. Collagen and elastin are proteins that provide the framework in animal connective tissues. And contractile and motor proteins, the last category, are proteins that are involved in movement. And this is probably the most common protein you think of. When you think protein, you think muscles, right? Muscle movement, muscle contraction is brought about by two classes of proteins, actins and myosins. And they're responsible for muscle contraction. Okay, now that we have examined all the different functions of proteins, let's build us a protein. Proteins, since they're polymers, since they're a biological molecule, are built by what type of reaction? Dehydration synthesis, or you may encounter it also as a condensation reaction. The way this works is that the carboxyl group of the first amino acid will be linked to the amino group of the second amino acid. And in the process, one molecule of water is removed. The bond which is formed between the two amino acids is called a peptide bond or peptide linkage. A dipeptide is two amino acids linked. Once you repeat the steps for the first two amino acids, you get yourself a polypeptide, which has many amino acids linked. Polypeptide. Here is a picture of a polypeptide with the peptide bonds in green. And what do you notice? Stop the video and take a look at this picture and think about it. What is it that you notice about the structure? Once you think you have an answer, you can resume the video. And if you answered that the basic structure of a polypeptide is the same, and the only difference are those R groups, you've answered correctly. If you look at this, this is the backbone in slight, I guess, blue-purple. It is identical. Every amino acid has an identical backbone, which contributes to the backbone of the polypeptide. And what really sticks out of this backbone are the different R groups or the side chains. The other thing I would like you to notice is that polypeptides have directionality. On the left, let's look at it as left and right. On the left of a polypeptide, you would have the first amino acid. And because the first amino acid contributes its carboxyl group to the peptide bond, the amino group is left free. So on the left of a polypeptide, we call this the amino end or the N terminus because the amino group is not engaged in a covalent bonding. At the very end of a polypeptide, you would have the last amino acid. The last amino acid contributed its amino group to the peptide bond. And its carboxyl group, and its carboxyl group is free. For that reason, the right end of a polypeptide is called a carboxyl end or the C terminus. And I want to show you again. The first amino acid will contribute its carboxyl group to the peptide bond. Therefore, its amino group will remain free. That's the N terminus. The very last amino acid is the amino acid that has contributed an amino group to the peptide bond, and its carboxyl group will remain free. That's the C terminus. N terminus, C terminus. Because polypeptides are built head to tails, head to tails, head to tails, they have a directionality. On one end, you have the amino group free, the N terminus. On the other end, you have the carboxyl group free, which is the C terminus. Okay, protein structure and function. What does it mean to be a functional protein? A functional protein will consist of one or more polypeptides. Occasionally, you have proteins that consist of multiple polypeptides. And these polypeptides have to be precisely twisted, folded, and coiled into a unique shape. The sequence of the amino acid will determine how a protein is going to fold and what its structure is going to be. And remember that the sequence of the amino acids is really determined genetically. And in the picture below, you will see a model, two different representations, two different models, a ribbon model and a space filling model of a protein. And this particular protein is called lysozyme. Lysozyme is an enzyme that is very rich in tears and saliva and mucus. And its function is to protect us from bacteria. It kills bacteria. And I'm going to show you another two pictures, models of structures of proteins. In the upper right-hand corner, what you will see is an antibody protein. And this antibody protein is bound to its target. And the target is a protein from the flu virus. So this antibody is directed against the flu virus. And you can see how the shape of the two proteins match and fit precisely. On the very left is an excerpt from a model that was recently built. And the model shows the SARS-CoV-2 spike protein, that's the spike protein of the virus that we've been battling, that is responsible for infecting cells. And it shows how this protein is bound to the ACE receptor. The ACE receptor is a receptor in our cells to which the virus attaches via this spike protein. So again, this is to illustrate how structure determines function. Let's talk a little bit more about structure of proteins. The structure of proteins has 4 different levels: primary, secondary, tertiary, and quaternary. And these are basically Latin terms for 1, 2, 3, and 4. So the first level, which is the primary, is the order in which the amino acids are arranged, or the sequence of amino acids. And the primary structure is determined genetically. The primary structure, in turn, will determine the secondary structure. And the secondary structure is held together by hydrogen bonds. Hydrogen bonds will cause simple folding of the chain. And the secondary structure, usually, there are small regions of the protein that will fold in a particular way. And then there will be regions that are not folded. Tertiary structure now, the third level structure, is much more complex. And it involves a 3D shape of the protein. And some proteins stop here. Others have quaternary structure. Quaternary structure when you have multiple polypeptides, 2 or more, that interact together to form the functional protein. Okay, so primary structure. It's the order of amino acids in a chain. The amino acid sequence is determined by the gene. It's written in our DNA, remember that. Even a slight change in amino acid sequence can affect the protein structure and its function. Even just one amino acid change can make all the difference. And we're going to explore why that is when we do some protein building and modeling. Secondary structure, shown here. It is local folding. And what local means is that some portions of the protein are folded one way, others are folded different way, and you also have these like regions in between the local folds that are unfolded. Again, this is folding along short sections of the polypeptide, and it involves interactions between adjacent amino acids. It involves hydrogen bonds. Hydrogen bonds are very important in local folding. They're weak bonds between what? An oxygen and a hydrogen in this case. and forms in sections of the 3D structure. There's two common secondary structures that we're going to learn about: alpha helix and beta plated sheets. On the left in this picture, you will see an alpha helix. On the right is a beta plated sheet. Okay, and here's a bigger picture of beta sheets and alpha helices. An alpha helix is a spiral, so it's easy to imagine. Beta sheets are a little harder to imagine, but if you take a piece of paper and fold it accordion way, you will get an idea of what a beta sheet will look like. And this picture does a pretty good job of explaining how beta sheets look like. How are these structures formed? Well, they're formed because hydrogen bonds form between the atoms of the backbone of the polypeptide. Now, it's important to understand that the R groups are not involved. They do matter. Why is that? Why is it that the R groups matter? I want you to pay attention here. See where the hydrogen bonds are in the helix. They're between an oxygen in the backbone and a hydrogen in the backbone of one amino acid, another amino acid. And an alpha helix is usually every 4 amino acids can form hydrogen bonds so that you form the spiral. But the alpha helices cannot form when there's certain amino acids there, because the R groups matter. Why do they matter? Just look at the picture, stop the video, and think about it. Think of this as a 3D space representation. Why would the R groups matter? Especially look at this R group here or this one right here. Why would it matter what type of R group there is in this particular amino acid or this one? And we'll get back to this a little bit later. Now the R groups here are highlighted in blue. Now I want you to ignore polarity and charge. What else do you notice? And if your answer is size, you are correct. The blue boxes, which highlight the R groups, are all different sizes, which shows that the R groups themselves, the side chains, can be a different size. So of course size matters. No one wants a small pizza, right? So let's look at this structure again. If this R group is too big, that it will not fit in this particular groove of the helix. So the size of the R groups does matter, even though the R groups themselves are not involved in this level of structure. So we are going to continue with this in the next video. For now, goodbye.